So this is going to be the biology marathon. Alright, so we don't have that much time. Let's begin with the revision chapters, which is chapter 3, which is the exchange in cells. The main thing you gotta know is about hypertonic, hypertonic, and isotonic solutions. Hypertonics are something that is dilute, and they have less concentration of those. Am I actually recording? Yeah. Okay. Less concentration of the stuff. You know what I mean? Basically, they have less concentration of so they have more concentration of water and they have less concentration of the other salt, like let's say sugar or something. Something that's hypertonic is more concentrated with that sugar, and that means there's less water. So there's less water that is being given, right? And in isotonic, both the amount of water and let's say something like sugar is the same. Okay, now you gotta know the next thing you gotta know is about osmosis. You should know that when there's a difference between water potential, the way it works is this, for those who have observed, is that you have to mention everything in water potential, where the reason why one goes in the other is because of the difference in water potential. And active transport is just the opposite, where instead of going from high concentration to low concentration, it goes from low to high against concentration gradient. So yeah, right. Now let's go to the next chapter, which is the bigger one, which is the leaf chapter. You gotta know this very important equation where CO2 plus H2O must equal to C6H12O6 plus H2O. And it's balancing as well. And here it is down here, you can see. Now, we gotta talk about what glucose is broken down. When we talk about when this happens, this glucose is used throughout in various processes. The first process it is used for is most importantly as storage in the chloroplasts of the cells. In the chloroplasts, all right, inside chloroplasts, it is used as fructose. It is used as used as storage inside the chloroplasts. The lichen, which is used, it also is broken down into lichen, and lichen is used for the xylem walls. All right, okay, and it also breaks down for cellulose for the walls of the vessels, and pretty much just breaks down into various parts which it needs. You can see right here, and it's also used in respiration for energy. Now, the leaf starch test. It's very simple, very basic. All right. So all we do is we boil the leaf. All right. We boil the leaf. That is going to be tested into hot water. Right. Into boiling water. This is to stop any form of metabolic reactions that is taking inside the leaf, and then heating it. As the heating it will damage the cell membrane. So allow iodine to go inside, and then we will add alcohol. And this alcohol, for a few moments, it will remove the chlorophyll. Then we will add iodine. If you see a color change, then there's starch. If you don't see a color change, then there's no starch. And for a variegated leaf, which is basically a leaf which has white spots, basically those white spots have no chlorophyll, meaning because if they have no chlorophyll, when you add iodine in, the ones with chlorophyll will change, while the ones that don't have chlorophyll will have no changes. Right, so what is transpiration then? Well, transpiration happens when, let's say, some amount of sunlight or heat takes away the water that is in the guard cells, or takes away water from the leaf itself. And what causes in the upper areas of the tree, right? So there is a difference in water potential between the top and the bottom. This one has less water potential, this is more. So it creates a suction system, like in basic diffusion. We know it just gets the water goes from the roots from high potential to low potential to the leaves. That's pretty much it. And this is determined by a few factors, which is humidity, which basically the more humid it is, the more slower the rate of transpiration. And we have humidity, then we have temperature. The higher temperature, the more faster transpiration will go. Okay, and then after that we have sunlight. We have wind speed. Sorry, we have wind speed. So wind speed is the faster the wind speed, the more the more humid, the less humid it becomes, and the faster the transpiration process goes. And of course, light. where more light means more heat too, so that basically means that it will be faster transpiration. If then between night there's less transpiration, so there's less light. Now, let's talk about xylem and phloem tube as fast as possible, right? With xylem tubes and phloem tubes. From what I remember, all you gotta know is that phloem tubes take sucrose, xylem tubes take water. Right. Now let's talk about, of course, the structure of the leaf. We start with the waxy cuticle. The waxy cuticle is there so it can absorb the sunlight. Then we have the upper epidermis. The upper epidermis, you notice, it has a nucleus, so that's important. But it doesn't have any chloroplasts. It doesn't have any chloroplasts so that it can absorb sunlight. But in the palisade mesophile, there are chloroplasts, and this is so that maximum sunlight is being absorbed for photosynthesis. Right. Then we have the spongy mesophils. These have small air gaps which allow for fast diffusion. Alright? Then we have the vascular bundle, which is made up of phloem and xylem tubes, which you already talked about, where phloem tubes transfer sugar, while the non-phloem tubes, xylem tubes, take water. And then below that, after the vascular bundle, we have the lower epidermis, which is the same thing, except the new thing is guard cell. The guard cell is important for the gas exchange where it shoots out oxygen and takes in carbon dioxide through the stomata, that is, in guard cell. Right. Now, that's that. There's also mineral deficiencies. Alright, all you gotta remember is that there is the magnesium deficiency, which makes the leaf more yellow, right? And then there is the nitrate deficiency, which basically reduces the growth of the plant. Okay, it doesn't really help it. Now, there's the limiting factors. These are the graphs for limiting factors. The limiting factors are temperature, light, CO2 concentration. And that's the three ones. What you gotta know is that the temperature graph I have a little drawing right here. You see the temperature graph is like an enzyme graph. It's basically the enzyme graph, where this is the optimum temperature, and everything below that is just denatured enzymes. Then we have the CO2 concentration graph. The CO2 concentration is in this sort of straight line and it's constant. The point where it becomes constant right here, this is called the limiting factor. The limiting factor of where everything begins. And here is where it's constant basically means all the factors become limiting. This is all you gotta know. It's the same goes for light. And this is the rate of photosynthesis. It increases and it just becomes constant due to limiting factors. Right. We're just speed running through as fast as we can, and that's pretty much the entirety of the plants chapter. Think about it. There's nothing else you gotta know extra. It's just this. And you just gotta understand it as well as you can, alright? So these are just the revision chapters. I'm going as fast as I can again, just speeding it all because we don't have time. Now we're left is the big ones, the ones that are in our final term syllabus, which will begin once I learn it a bit,